For 2 decades, regenerative medicine asked a single question about human fat: how many stem cells can we pull out of it? Adipose tissue is one of the richest reservoirs of regenerative cells in the human body. The clinical instinct was straightforward: extract those cells, concentrate them into a high-density dose, and inject them. But that approach carries a significant biological cost. The act of extracting a cell rips it away from the physical environment that tells it what to do. A different priority is taking hold in the field. The focus is shifting away from how many individual cells you can extract and toward how much of the living tissue you can keep structurally intact. Raw cell count is a misleading metric. True regenerative potential relies on preserving the intricate architecture that actually makes those cells function. Fat is not a passive energy storage depot. Inside it sits a highly organized living microenvironment called the stromal vascular niche. Picture fine capillaries threading between the fat cells. Wrapped tightly around these capillaries are parasites. These cells sit directly on the vessel wall, acting as first responders that activate into repair-driving cells when they detect tissue damage. Woven through this space are telecytes. They extend extraordinarily long, thin projections, forming a 3-dimensional communication web across the entire tissue. Held quietly within this network are stress-enduring cells. They remain dormant, resting until a specific injury signal arrives to wake them up. Binding all of this together is the extracellular matrix. This physical scaffold uses its own stiffness and structure to send specific behavioral instructions to the cells living inside it. None of these elements work in isolation. The niche is a coordinated system. Biological function is an emergent property of this exact arrangement. Cells in the right place, next to the right neighbors, embedded in the right matrix. Processing this tissue comes down to 2 opposing philosophies, and the method you choose dictates the biological product you end up with. The first method is isolation. Whether using chemical enzymes or heavy mechanical force, the explicit goal is to break the tissue down to separate out the individual cells. The act of separating cells is inherently destructive. Capillaries are torn apart, the delicate telecyte networks are severed, and the instructive matrix scaffold is dissolved. What remains is a stromal vascular fraction, a disorganized cell suspension completely disconnected from its native instructions. It delivers a one-time bolus of isolated cells. But without the architectural support of the extracellular matrix, most of those cells lack the environmental cues required to survive and function. The second philosophy is preservation. Instead of tearing cells out, this method gently reduces the size of the tissue while explicitly protecting its internal architecture. Liposmart is a medical device engineered entirely around this tissue-preserving philosophy. It processes adipose tissue through progressive polymer membrane filtration. Passing gently through membranes of 1,200 and 700 micrometers, the tissue retains its structural integrity without crushing or spinning. Throughout, the tissue remains constantly bathed in fluid immersion, buffering fragments from mechanical stress. The system is completely closed-circuit, sterile, and single-use. The procedure utilizes only physiological saline or Ringer's lactate, with absolutely no enzymes or harsh chemicals. This point-of-care procedure produces micro-fragmented adipose tissue, or MFAT. It is a fluid suspension of small, self-contained pieces of living tissue, rather than a bare cell suspension. This chart shows the biological impact of preservation. Published characterizations show that micro-fragmented tissue retains roughly 34% of its parasites, compared to just 8.5% in enzymatically isolated fractions from the same donors. But it goes beyond raw cell counts. The extracellular matrix remains intact as an instructive scaffold. The telecyte communication network stays in place, and the stress-enduring cells continue resting in their native, unactivated state. This preserved architecture allows the tissue to maintain its secretome, the natural, ongoing output of extracellular vesicles and signaling molecules. Characterization of this secretome has identified hundreds of vesicle-carried microRNAs. These specific molecules have documented roles in steering local inflammation towards resolution and actively protecting cartilage. An isolated cell injection delivers a single rapid pulse of biological activity, after which the vast majority of those cells die. Preserved MFAT acts as a slow-release biological engine, a small piece of living architecture that continues to signal into the surrounding tissue over time. The Liposmart platform applies this exact preservation biology across 3 distinct chamber sizes, each matched to a specific clinical demand. The 60-milliliter model is built for small-volume fine work, including facial aesthetics, the scalp, scarring, and delicate periorbital treatments. The 120-milliliter device is the versatile flagship platform. It covers a broad range of procedures, spanning orthopedics, sports medicine, gynecology, and reconstructive cases. For high-volume orthopedic and massive reconstructive applications, the 275-milliliter model provides the necessary scale. Different volumes, one shared principle. The platform ensures complete tissue architecture is preserved at every scale. Micro-fragmented adipose tissue now has a rapidly accumulating clinical evidence base, with the strongest data currently found in knee osteoarthritis. The results across global trials are mixed rather than perfectly uniform. This is the hallmark of a serious, evolving scientific field, where researchers engage with the trials that show benefit alongside the ones that do not. Liposmart's own device-specific clinical validation is actively building on this established biology. This direction is led by the platform's scientific director, Professor Carlo Ventura. Serving as a former editor-in-chief of the World Journal of Stem Cells, his foundational research in mechanobiology established the scientific rationale for preserving the stromal vascular niche. Because it processes tissue mechanically without enzymatic digestion, Liposmart operates as a point-of-care CE Class IIa medical device, manufactured entirely in Italy. Don't extract the cells. Preserve the living tissue that makes them function. To preserve, to regenerate, to heal. This is Liposmart.